We'll go to Slido for questions, so a reminder to please not only add your questions there, but vote, and we'll go off of there. So we'll start off the top here, Christian. So this says, great to see the repeated examples of learning from the one epidemic to be prepared for the next one. So what are your thoughts on wastewater sequencing and the ways you might incorporate that into your surveillance system?
Yeah, I do think that water surveillance or wastewater sequencing actually have a lot of promise. And I strongly believe that the future of surveillance actually will be through wastewater. The reason being, we do have some clear evidence from our setup, our labs, and then also from the work that we're doing that, through wastewater, you actually uncover a whole lot and then a whole lot ahead of time. I basically just want to give you a very small example, the way we actually use that in our small campus, actually to help even our university management to respond to COVID. So when COVID started, then we had students on campus, for instance, and then we had them. So we assigned a few of our staff to go around and then get wastewater, within the campus at different time point, geolocated. And we basically were seeing, even though we do all of these tests and everything, the levels were high up. And then that resulted in when they said, oh, well, you know what, we're dealing with an issue. If you keep having it, it's a time bomb. So we sent the students back home. And as they went back and then collect samples at all the different time points. And then you see, COVID, level in the wastewater crash down. After a few, I think it was 2 or 3 months, and they were told, we're reopening, and then the students are coming back. They come back 2 weeks after. We get back and collect the sample, then we are back up. So I think that, and then apart from that, through metagenomics, you see a whole lot of stuff in water. AMR, and then at some point you see, monkeypox, and then a whole lot into wastewater. I think it's really great, a great way to do it. But I think it's also the fact that, we just have to be very cognizant. You need to adapt this approach to wastewater to the environment where we are. In the US, it's easier because you have, waste treatment plants, right, where you could go and then get samples. But in the context of Africa, you don't have, you don't have waste treatment plants. So you need to think about how best you can actually adapt that to the environment. Yeah.
Excellent, thank you. The next question is really around global data sharing mechanisms. Just as an aside here, we in Canada were involved in the same at Genome Canada, we're involved in sort of the genomic surveillance of variants of concern and so on. And Christian was a real voice globally for improved data sharing processes, data access processes. So this is a really relevant question here. So I wonder if you could speak to your opinions around how well existing those global data sharing mechanisms or processes are supporting or are not supporting future pathogen preparedness, and what sorts of improvements would you like to see?
Well, I do think that, global data sharing is something that, if you look, many people are willing to. But the reality is, it's also somehow lead to exploitation if care is not taken. I think it's important going forward to think about setting up some guidelines and set up some regulatory framework that will encourage people to share data. And those regulatory framework should really include the whole aspect of benefit of data sharing. It makes no sense for people to share data, for instance, and then you have some big farmers getting the data and then go and create vaccines, and at the end of the day, they tell you that, you can't even access the vaccine. So I think, I'm not against data sharing. I'm not against the fact that people shouldn't use the data to advance science, but we also have to be sensitive to the fact that while people share the data, but then you need to give back. How do you give back? I think there's a need to think about, can you encourage people that share the data, they don't have enough resources to give back, in terms of maybe technology, trips, in terms of IP, yes, in terms of, making these vaccines affordable. It was very frustrating for Africans, I can tell you, because we were championing data sharing, but at the end of the day, you see all of these big pharma creating vaccines, and even with money, we didn't have access to vaccines. And that is not acceptable. And that was a major blowback and a major setback to data sharing, because I can tell you that at some point, many countries said that, you know what, we are not releasing data anymore. Apart from that, you also have the political discrimination that came out of it. You report a disease or you report a pathogen for the global health benefit. But at the end of the day, you attack, your country is attacked. You are banned from traveling. That is really a major disservice to public health. And we got to change those kind of things. We got to shy away from those kind of attitude. But then the question is, how do we set up a framework where what I call the vagabond nations are held accountable? It doesn't make sense to create a global health community and then think about that people are sharing, and at the end of the day, some people take these things and they make theirs. And and at the end of the day, you that share the data, you are completely at loss. So I think there's a need to create a global framework. But how do we hold people accountable? That's the question.
Yeah, such an important message. Thank you for sharing that. The next question gets around the sort of the linkages you mentioned here with the zoonotic origins of many of these viruses, and what the key challenges and obstacles maybe being that you face when implementing some of the genomic surveillance sort of processes in West Africa around zoonotic sources.
Well, I do think that right now, it's still what I call in its infancy. So, and then it is important to create evidence. It is important to create use cases. And we have a few use cases now to demonstrate that it's important to do zoonotic research or to use a One Health approach, that it involves zoonotic research, that zoonotic surveillance, environmental surveillance through wastewater, for instance, and also involve, clinical surveillance to humans or community-based surveillance. So these are things that are really needed in order to, to understand the value of this. But there is a need for use case. I've come to learn over the years that, we can talk from morning to night. We can go and then publish whatever we want to publish in all these great papers and everything. These are academic exercises. When it comes to implementation, when it comes to doing the work and then taking the decision, it's important to actually present the information and use and present use cases. Use cases are what convince people to take the decision in the direction that you would love to see as a scientist.
Yeah, thanks. I think that's so important, in a way where the work you're doing in West Africa can really be a lead for the same sorts of challenges we're facing all over the world. And that's the intersection of how the technology intersects with the environment in which a lot of this work is happening, and that's a challenge many of us are facing. This is a very Global Alliance question around standards. Thinking about what is the role that standards play or could play in cases like the ones that you've presented here today. So how can sort of this community support the work in pathogen surveillance that you described?
I think partnership and collaboration is very important, and then through partnership and collaboration, we have set of standards. Can we do things the same way? Can we use the same, approaches that are a bit more standardized in a way that the data are somehow comparable? So I think it's important to think about how we could set up some guidelines, some set of rules, and also creating what I call access. It's good to set up rules, set up standard, but the problem often I would say is how do we create access for people to meet those standards, and then it's not just to meet the SOPs and everything, but in terms of meeting the reagents, to meet everything. So it's important to think a bit globally about it, and then also importantly is actually, one of the major limitation for setting standard is going to be cost, right? And then we keep thinking this all the time. Unfortunately, I won't be here tomorrow, but I think I should have been here on Saturday to present about GENCO that Nikki and I and then Arbour are driving. One of the things, you have a great initiative, but the question often is that for implementation, how do you get this implemented in a way that can help everybody to add value? But the major limitation is often, for us in the developing world, cost, because of all of the middlemen that you have in between. You buy a reagent in the US for $100. I buy it for $500 in Africa. How do we contribute? How much can we contribute to that? But it's simply because of middlemen in between. But can we find a formula that can make this reagent more affordable so that we can actually just go back and start using all the different standards that you're talking about in terms of data infrastructure, architecture? So do we have the same kind of standard? How do we build, the architectures in a way that we can have the data that are shareable in a way, or stored in a way that that we can all access? Shared, we can all share. So I think there's a lot of things that we need to do in terms of, in terms of building those kind of standards.
Yeah, and maybe there's a link back to the conversation around data sharing and benefit sharing, and maybe cost sharing should be included in that as well, that if we're creating global resources with global standards, that we need a global approach to making sure that we're actually getting good representation. I think that's a really valuable message. This is, I think, a really great question. I think all the questions are great, by the way. Sorry, I don't mean to privilege any of them. But I think this is something that we think about a lot as well in terms of the aftermath. Following the COVID pandemic, where there was so much justifiable attention being paid to pathogen surveillance, to building out tools, to investing in capacity building, and so on. As we turn our minds to other things, perhaps, how do we maintain the momentum? What is the aftermath, in effect, around data sharing in Africa and globally, frankly? In the end, do you think that we will have learned something from the pandemic that allows us to continue to invest in the necessary infrastructure for this kind of work, or are things already starting to revert back to how they were pre-pandemic?
I mean, you don't need to be a genius to understand that we're going back the way pre-pandemic, the behaviors are changing. We're back, and unfortunately, we just have to think about doing things differently. And then probably one of the ways to do things differently is actually to start rethinking the way we're doing science, or rethinking the way we're collaborating. And I think co-creation is going to be very important to build trust. And then also there's a need to do what I call long-term investment. To invest long-term. Parachute science is not going to help. And in parachute science, again, it's the fact that you want people to say, okay, now, before, there used to be a whole idea of collecting samples and then doing it in some lab. Now it's getting difficult. It's like, let's submit all the data somewhere. We need to create trust, and then we need to let people understand that, even when they are sharing data, they still, can benefit from those data in a way that the communities where they get the samples, could trust them. As a scientist in the developing world, I'm going to the community. I'm telling them that I want to do this great work. There's a potential for future benefit and everything. And I go back there 5 years down the line, they ask me the question, so what did you do with this sample? 10 years down the line, a little kid that was a year, he's 10 years, and he sees me on the way, and he said, look, that sample that you collected from me, what happened to it? So I gotta be, I must have that information. I should be able to tell him with confidence that this is what is happening to it. This is what your sample contributed. It probably also contributed to developing a vaccine, but then the question is, can I have access? Do my junior brother have access to it? If we can set up a framework that will help people to have confidence in sharing because they can have benefit in sharing, it's going to be very difficult. And then mind you, we don't have that much data, in terms of human genome sequences from Africa into the global database. How are we encouraging people to actually make sure that we make those data available? How are we encouraging people to actually generate so much data? And then the question is, after the data are generated, what benefit do they have in return? I think we need to rethink the way we're sharing the data, the way we're collaborating, and then be more inclusive, and then be more pragmatic in the way we want to basically just give back.
Awesome. Okay, we could do this all afternoon, but I'm afraid we have another great session. Congratulations on a phenomenal body of work so far. Best of luck on the continued work. Please join me in thanking Professor Happi for this phenomenal presentation.
Thank you.
Okay, so we're going to keep things rolling here, and we're going to go next to a panel discussion. This is a discussion titled Towards Inclusive Research, Diverse Datasets and Cohort Representation. Up first is Maili Raven-Adams, and Maili is known to many of you as she co-led work in the Regulatory and Ethics Workstream here at the Global Alliance for Genomics of Health. She now works as a researcher at the Nuffield Council on Bioethics in the UK, focused on bioethics and policy. She holds a law degree from the University of Glasgow, a master's in medical law and ethics from the University of Edinburgh. Maili, it's all yours.
Hi, everyone. Hi. Is this not on? There we go. Okay, I'll start again. Hi, everyone. It's nice to see so many of you again and to come to San Francisco and see so many familiar faces and also new ones as well. As I've just been introduced, I now work at the Nuffield Council on Bioethics, but today I'm putting my old GA4GH hat back on and I'm presenting in that capacity rather than representing the views of the council. So in this presentation, I will be presenting the policy that's been developed by the Diversity in Dataset subgroup of the Regulatory and Ethics Workstream, which is on considerations for how genetic and genomic researchers should approach thinking about diversity in data. This has been a huge group effort and has been co-led by Anna Lewis and Tina Hernandez-Boussard, who couldn't be here today, unfortunately, and myself before I left in May. The policy itself has gone through multiple iterations due to being reviewed by the subgroup, by REWS, and also it's been out for public comment as well. Earlier this week it was reviewed and passed approval at REWS, which is really good news, and we'll be going for a steering committee approval hopefully later in the year to become an official GA4GH deliverable. The current version you can find here on the QR code if you want to have a look while I'm going through, or just have it afterwards. I'll put it on the last slide as well. And throughout this presentation, I'll be going through the issue that we outline and also how we want to approach that issue through the framework that the group has developed. Before moving on, I just want to say a huge congrats to the group for pulling this together. I haven't been as involved as I would have liked over the last few months, and it's really come together, and I think it's a really good contribution to this space. And I also want to say a special thanks to Beatrice and to Dia as the REWS contributors for picking up where I left off before I left. I just want to also say that this will be quite high level just due to time. So there's lots of really good examples in the text itself, so please go away and have a look at those afterwards. So as I said, in our policy, we spend some time outlining some of the background to the issue that we try to address. So firstly, there are many, many different calls for diversity in data set out there, all aiming to ensure that the benefits of genetics and genomics can be realized for everybody equitably and reliably, without perpetuating the health inequalities that we have today. For example, many of you will be really aware of the commonly cited statistics showing that 86% of all genomic studies were conducted in those of European descent in 2021, which obviously shows that the genomic research that we're doing does not have translatable findings for absolutely everybody. However, despite the importance of these calls for increased diversity in genomic datasets, we recognise that there is a lack of specificity about what is actually meant by diversity and how we can achieve this, which leaves a lack of clarity for researchers who want to address these calls in practice. Secondly, there's often a conflation between concepts like diversity and representation. And although that we agree that representation can be really important, and we certainly don't have enough of it right now, there may be good reasons for a lack of representation. For example, to maximise some genomic projects, oversampling individuals from recent genetic ancestries from Africa, for example, compared to the rest of the world, may be needed. Thirdly, there are also multiple dimensions of diversity that it might be important for researchers to consider. These obviously include genetic ancestry, which is what many of the calls for diversity in data are focused on, and other factors researchers might want to consider are sex or environmental factors, such as the social determinants of health, like geography or whether somebody smokes or not, which can outweigh factors including genetic predisposition. In each case, it's really important for the researchers to think about, what concept of diversity is important for them and get it right for each given project. Fourthly, the concepts and terms that we use matter and have ramifications that we need to be aware of. There can be problems with putting people in categories which might happen when we're thinking about how to increase diversity in data sets. We need to remain cognizant that the birth of genomics and genetics are intertwined with that of eugenics and unfortunate events to try and justify racism. When looking at increasing diversity in data sets, there are dangers of reinforcing the false notion that humans come in distinct biological types. Lots of the calls to increase diversity in data sets focus on increasing ancestry. However, without further clarification, this broad term suffers from ambiguity that might be harmful in practice. For example, empirical work has shown that some researchers use ancestry as a distinctly biological one developed by genetic analysis, while others use ancestry merely as a social phenomenon. And the problem with this is that it can allow for the conflation of social and genetic categories, which can also lead to ignoring the impact of environmental factors that might be important as well. And so we include within our policy that researchers should use genetic ancestry, if that's what they mean, rather than just ancestry. And researchers should be transparent about what is meant by the categories they adopt. And lastly, one size does not fit all, so we can't provide one definition of diversity that will work for everybody. The context in which researchers are working vary substantially, for example, based on cultural practices, the economy, where they're based, and whether the research is embedded in healthcare or not. Because the goals of each research project will be different, the types of diversity that matter will vary for each research team as well. So, in our policy, we say that it's not enough to simply call for diversity within datasets. This needs to be done in a way to understand what types of diversity and why and how to act on this understanding throughout the research process for each research team. So the policy itself tries to address some of the issues that I've outlined so far. And the subgroup set out this framework, which you can find on the slide, and I'll go through in a moment, to kind of help researchers address the question of what types of diversity are important for their own research and how to achieve this as well. So before I get into explaining that a bit further, we provide some key messages that frame the whole framework in practice. So first of all, we recognise that there are complex reasons for the lack of diversity in data. This includes a well-founded lack of trust in genomics and genetics research based on instances of exploitation, cultural ignorance on the part of researchers, a fear of genetic discrimination, and existing barriers to participation such as transport costs, childcare costs, language barriers, and many more. And helping researchers achieve the benefits of more diversity involves prioritising transparency, accountability, culturally appropriate behaviour, as well as recognition and respect for the role of marginalised communities participating in research. So secondly, we say that we uphold a mandate for inclusive practices, and so that researchers should make it as easy as possible for differently situated people to participate in research if they wish to do so. This can include respecting community practices and data sovereignty or governance, as well as making considerate choices about the language that we're using. Oops, go back. Gone too fast. We also highlight here the importance of data sharing, which obviously is what lots of the talks here at GA4GH Plenary have been about. So we recognise that across genomic medicine, research results are optimised by integrating data from as many individuals as possible. This can be done by sharing data, which can in turn contribute to increasing the diversity researchers can access in the available data to them. However, data sharing needs to be done responsibly, recognising human rights, other legislation, and ethical guidelines which may limit what data can be shared. And lastly, we keep at the top of everything that equity is often the most important concept when determining what dimensions of diversity are important and how to achieve that in practice. So I'll move on to talk a bit about the actual framework itself. So the policy itself first outlines ways researchers can understand what types of diversity in data are important for their projects, keeping in mind that diversity in data is often a means to achieve ethical ends. So researchers should take time to clarify the goal or goals of their project and consider how these align or fail to align with ethical norms such as public benefit, equity, justice, fairness, as well as fundamental human rights and the improvement of healthcare. To achieve this, we include that researchers call on bioethicists, lawyers, and local communities that will be impacted by their research to support them in this process. And after following that, researchers should then ask themselves, why is diversity in data important to achieve those goals? And this should include consideration of who should be in the data and what data points are the most important to know about them. Secondly, the policy outlines a systematic way for how researchers can achieve the types of diversity that they've identified as important for their project. So armed with some degree of clarity about the types of diversity in data needed to achieve the project goals, strategies will need to be adopted consciously throughout the data lifecycle. And this lifecycle you can find on the slide here, but we recognise that each point on that lifecycle might not be relevant for each research team. And the main point from this part is that to achieve diversity is not just about who is in the sample. Considerations include thinking about the implication of choices we make throughout research entirely, and how to encourage benefit sharing, including building research capacity, equitable collaborations, and avoiding helicopter science. This benefit sharing needs to be done responsibly and be developed with research participants and local communities with awareness of where some benefit sharing may be illegal. So we provide some considerations that might help or guide researchers. I've just put a few of the examples here on the slide. There are many more of them within the text itself. And this involves including reference to just ancestry, like I've said, and ensuring there are attempts to identify data sets that adequately capture the different dimensions of diversity that are identified as important for research teams. This data should be collected in a way that is culturally and contextually appropriate, with barriers to participation being identified and reduced to recognise the value that participants bring to our research. Another example that I've put there at the end is at publication stage, where researchers should carefully state how results are expected to generalise or fail to generalise. And here we highlight that research that centres around diversity in data can bring too much attention to the small amount of genetic variation that might divide us, and it can be used to tell oversimplified stories about human groups and can contribute to stigmatisation of already marginalised groups. So these are just some of the things that we think is important to keep in mind, and the whole framework itself as kind of a way for researchers to be transparent, thinking about their processes, and also being transparent outwardly for others to kind of review their work, and for instances of like reproducible research. So I just want to highlight as well, I don't have time to go into them, but in the text itself, we provide 2 examples that work through the whole policy so that people who want to use it can have a better understanding of how it might apply. So we've got one reporting, looking at minimizing reporting false pathogenic variants, and another one on the clinical impact of differential predictive performance of polygenic scores. So I've put the QR code there again if you guys want to take a snap, and you can look through it later on. And although we have a panel now, if you've got further questions, please feel free to email myself or Anna or the REWS team as well, and we're here to help. Thank you.
Excellent. Congratulations to you and the REWS panel. That's really impressive work. You can also put your questions on Slido just to remember. Up next, we have Alham Saadat. Alham is a scientist and ethicist and the Associate Director of Scientific Equity at the Broad Institute in the Inclusion, Diversity, Equity, and Allyship Office. Alham, the floor is yours.
Thank you for the invitation to speak today, and it's been wonderful to hear everyone's fabulous talks and learn about a lot of the important work that's happening across GA4GH. Today I'll be talking about Broad's multidimensional approach to inclusion and equity in biomedical research. I'll start with an overview of our holistic approach to thinking about DI efforts across the Institute broadly, and then go into how that's related to specifically how we think about equity and inclusion in the research itself. Broad's DI office is comprised or is led by our Chief Equity Officer, and the work is separated into three different focus areas. The first focus area is really thinking about how to create a community that and culture within the Institute that is really supportive of everyone that is there, where everyone can feel that they can belong and thrive in the various ecosystems that they work in. The work that I lead is focused on the research itself, so it's really thinking about how we can develop and implement strategies to make our science inclusive and equitable, really ensuring that the benefits of biomedical research are shared by all. And I'll dive into what that means in more concrete detail throughout the talk. And the last focus area is scientific workforce diversity. So this is led by my colleague Giselle Velez-Ruiz, and these efforts are aimed at ensuring that we are doing a good job of increasing pathways for scientists who are currently underrepresented across the scientific ecosystem. And I just wanted to mention, these are three distinct focus areas, and we conceptualize them as separate but very interdependent. And the success of each of these actually is very closely related to the success of the other domains. So drilling down a little further, what do we mean by the work that would be involved in terms of scientific equity? Thinking about it, the work aims to increase understanding of how the decisions we make at all stages of a research project will impact health outcomes for people, particularly people that have been historically excluded, underserved, or harmed. And the work of scientific equity falls into four kind of focus areas or big buckets of work. We aim to foster learning and discussions across the Institute, develop new bioethical capabilities and increase bioethical engagement across our ecosystem, develop and share educational resources, good practices, and toolkits that can enable folks to better integrate some of these principles into their work. And build collaborations with organizations engaged in similar work, both within the Boston-Cambridge area, but ideally across the US and global populations. And now I'll focus on a few of these, given the limited amount of time for this talk. The fostering learning and discussions. We've created an equity in biomedicine seminar series. The aim of this seminar series is to focus on critical issues related to equity and inclusion in biomedical research, really getting our speakers to engage with our community on the ways that their work directly impacts health outcomes, health inequities or inequities, to really drill down into who will be affected by the work, how will they be affected, and why. Some of the past seminars that we've hosted have covered these topic areas of sex and gender minority inclusion in research, thoughtful use of population descriptors in genomics research in African diaspora populations, research is as relational, really thinking about the role of community engagement for equity in precision medicine. And if anyone is interested in viewing any of these past seminars, they're recorded and publicly available on our Equity in Biomedicine YouTube channel that's hosted by the Broad. Upcoming seminars include thinking about increased genetic diversity and the needs of that specifically in the context of cell models and CRISPR experimental tools. Population genetics and the responsibility to counter nefarious use of research. And then we're very excited to host the NIH's Director of Minority Health and Health Disparities, Alessio Perestable, early in 2024. The previous seminars have all been kind of focused for the Broad community. Prior to the recordings, future seminars will be open to the public, and I'd be happy to share information about how folks can join for these upcoming sessions. Thinking about building on ways to have our community engage in learning more about how their work impacts various communities. We're also thinking about how to address bioethical needs that go bioethical thinking about compliance in regulatory domains. So we're building new bioethical capabilities and aimed at increasing bioethics engagement across the Institute. And through the Broad Bioethics Initiative, this is an initiative that will go live early next year. The focus of the work will be to support bioethic domain beyond regulatory domains and domains that require collaborative problem solving among administrative compliance functions, ethicists, and scientists. It will aim to help produce better scientific research through, for example, clarifying concepts and helping align both scientific needs and ethical ends, and producing leading and impactful bioethics scholarship. So Broad leads in science and technology development. It has the opportunity here to also play a role in leading in considerations of how these developments can and should impact people's lives. So really thinking about the idea that with the privilege to work in cutting edge research comes great responsibility. This work is co-led by Broad colleagues Anna Lewis, who's one of the champions of the diversity and datasets efforts at GA4GH, as well as Rosie Hosking, who leads efforts that are focused on the biology of trauma initiative at the Broad. The Broad bioethics work is, here are some examples of the types of domains that we could potentially be useful to our community. Kind of thinking through return of results and benefit to research participants in their communities, bioethical considerations when increasing research participation from underrepresented groups in under-resourced settings, so thinking about both domestically under-resourced settings and also low and middle-income countries globally. Considerations of what types of diversity matter for what types of genetic research, what Maili just eloquently spoke on. Responsible data sharing beyond regulatory and compliance domains, and thinking about bioethical issues surrounding artificial intelligence and machine learning, as we've had some great talks earlier today about how that field is quickly evolving and some of the ethical opportunities and challenges that come with that. The vision for the BBI involves 3 strands of work. So we aim to offer educational materials to our community that allow them to better identify ethically relevant features in their work, and also engage with those issues in a meaningful way, and also provide educational materials that take a deeper dive into particular topic areas, such as scientific racism and some of the other topics that I just previously mentioned. We're also going to launch a bioethics research consultancy service in early next year. This will be available to all Brodies, and it's aimed at providing useful, practical advice on issues raised in various bioethical domains. And then the third strand of work involves projects. So these would be initiated by Broad scientists in collaboration with bioethicists, aimed at producing state-of-the-art practical bioethics advice and scholarship. And I'll finish here by just kind of circling back to the approach to DEI and thinking about equity and inclusion in research involves not just a consideration of directly the research itself, but really thinking about these additional components of culture and community, ensuring that we're creating spaces for researchers that are healthy, that sustain cultures that are thoughtful of identifying resources for good mentorship, that are supportive of each other, and that are overall really cognizant of creating healthy ecosystems. In addition to that, really critical to think about, in addition to thinking about scientific equity, it's really important to think about who's doing the work and making sure that there's a better representation for groups that are currently massively underrepresented across STEM fields. So efforts to really think about increasing pathways to expand and accelerate the diversity of future scientists is a really critical part of how we conceptualize inclusion and equity in our research ecosystem. And I'll end with acknowledgements of the amazing teams that I get to work with, our inaugural and amazing colleague and Chief Equity Officer, Renee Salazar, who unfortunately we lost about a year ago. Suddenly, our new Chief Equity Officer, Kedrick Perry, and the other associate directors in our team that are leading really important work across the other domains that I described, as well as the co-leads for the Broad Bioethics Initiative work, Anna Lewis and Rosie Hosking. Thank you.
Thanks, Alham. That's really impressive work. Thanks for sharing that overview of the BBI. Look forward to hearing more about it during the Q&A. Up next is Calvin Ho. He's an associate professor with the Faculty of Law and co-director of the Center for Medical Ethics at the University of Hong Kong. Calvin.
Thank you so much for the kind introduction, and I'm grateful for this opportunity to weigh in on the discussion and also to contribute to the diversity in dataset policy. So let's see, I'm going to get the slides up. Here it is. So I thought I'll take this opportunity to invite everyone to just zoom out a little bit. We've heard about the wonderful policy that's come up on diversity in data sets and potential application of this particular policy. This invitation now is really to think again more broadly about what exactly we're aiming at. And my argument is that it's directed at, from an ethical standpoint, advancing the goal of epistemic justice. So I should explain a little bit about what this means from an ethical standpoint. And I also think that this is, with the benefit of this policy and also related developments, a great opportunity to weigh in on some of the important developments that's currently happening post-pandemic, and one of which relates to the pandemic treaty that's currently headed by the World Health Organization. So I think this is an important opportunity to learn from the experiences that we've gathered and to hopefully advance to a better place, having heard from Professor Happi, right, some of the things that we could subsequently apply to put us in a better position particularly where R&D capabilities are concerned to respond to the next pandemic. Of course, as we know, sadly, this will not be the last time we will see a pandemic. So, so what exactly is epistemic justice? As you see, it's being addressed to a degree in the diversity in datasets policy. And here, of course, is what's most significant will be its emphasis on equity. And I'd like to relate this in particular to inclusiveness. So when we think about the sort of work that's being done, to what extent are we being inclusive in terms of who gets to contribute to the advancement of this knowledge, and ultimately who do we count as putative members of an epistemic or knowledge community? So have we excluded certain individuals? Have we excluded certain approaches? And why exactly have we marginalized these particular individuals and approaches? So as you see, if you go with a number of ethical concepts, I just refer to one of them, and that's the framework that's developed by Miranda Frinker. So for those who are familiar with this notion of epistemic justice, you'd see that there are two different forms of epistemic injustice. So one of which relates to testimonial epistemic injustice, or the hope of advancing testimonial epistemic justice. That's flipping it the other way. So what testimonial epistemic injustice relates to will be when individuals themselves feel that their contribution will be undermined essentially because of their social standing or their social position. So this, of course, from our earlier discussion, if we wonder why is it that certain marginalized groups or members from marginalized communities feel that they're not coming forward, so they might feel that whatever contribution that they make, the testimony that they produce, would ultimately be undermined, or it could be used in ways that would ultimately lead to greater discrimination or stigmatization. So obviously, given these sort of challenges, you'd not find individuals coming forward. So it could be individual, it could be at a systemic level, which is what epistemic justice in a different form would speak to. I'll get to that in a bit. But just to get us thinking about this particular point, there would have sociologists telling us that where you'd have researchers working in a more localized space, their work may not be sufficiently recognized as contributing to a field only because it's a more localized space. It's not considered generalizable knowledge. So this is one example of how perhaps particular matters, particular approaches in research may not be given the sort of recognition that they deserve. So that, of course, speaks to one form of epistemic injustice that we're invited through the policy document to start thinking hard about. And of course, getting back to the structural concern, whereas the earlier form, the one form of testimonial injustice are essentially individual in nature, hermeneutic epistemic injustice is always structural. So, and I think this is especially important to consider from an epistemic standpoint. So when we have the sort of data that we create within knowledge field, ultimately, sorry, the arrows in the last point are kind of off the mark, but here you'd find, if we think about the sort of data that we have, ultimately, does it speak to social diversity? Of course, we can debate whether social diversity is a necessary good, but if we agree that social diversity itself is a good, and it can contribute to knowledge development and serve as a check to biases within a particular knowledge system, then ultimately, that's one way to think about the sort of data that comes out of the research endeavors that we have taken on. Ultimately, when we think about social diversity, the sort of data that we have, do they encourage cognitive diversity? Do they speak to multiple types of data or evidence for that matter, and ultimately applied in a form that would support theoretical and evidential robustness? So these may not be considerations that would ordinarily be taken on within a particular initiative, but ultimately I think if we take a step back, think about what this new policy is directed at, I think this is an important goal that it encourages us to take on. Because ultimately in any kinds of knowledge discipline, as you know, one of the key concerns would be shared reality bias. So from a philosophical standpoint, whereas you'd have Thomas Kuhn, as some of you may be familiar with him, he argues, I think, quite on very strong grounds, essentially, that the scientific endeavor should be protected from unwarranted intrusion. But then we would have Karl Popper reminding us that ultimately science must operate on the basis of skepticism, informed skepticism, in order to serve as a check to the sort of biases that we may be concerned with. So the last bullet point, if we think about it in a public health kind of context, sorry for the grammatical error there. So public health workers themselves are not immune from prejudicial forms of reasoning because they are ultimately socially situated agents. So they are, or they would operate within the social environment. And this, I would argue, is no different from researchers within any research community. So what I think the new policy, the guidance document on data diversity seeks to do is essentially to reintroduce some of these checks in place to encourage researchers to be critically reflective of their enterprises and to see ultimately whether their endeavors, quite apart from advancing the knowledge goals within an epistemic system, also could speak to some of the epistemic injustices that we find either at the individual level or at a broader structural level. Well, here's another example, I think, of a group that attempts to put in mechanisms to advance the concerns of epistemic justice. So switching gear a bit from the sort of epistemic level engagements that we find, I think it speaks to, of course, ultimately how we include individuals, how we include different approaches within the knowledge system. And this inclusiveness, I think, is important as a link to some of the developments that we find in global health, particularly where the pandemic treaty is concerned. So here, of course, in this paper, you'd find that it's linked to a number of very critical concerns that we've heard from our keynote speaker for this particular session, how it ultimately would link to trust and engagement of individuals to participate and to contribute, not just to public health sort of goals, if we think about it within the pandemic context, but also to co-creation of knowledge. And this we may not see enough of, especially when we think about marginalized individuals and communities. So here, if we look at the zero draft of the pandemic treaty, or otherwise the pandemic accord, depending on what form ultimately this treaty will take, you see that there is a clear section on R&D, and of course R&D speaks to a number of points that the keynote speaker have already spoken to. It's less explicit in terms of the need to ensure, for example, data diversity and have mechanisms in place that could ultimately advance epistemic justice. But as we've seen in the past pandemic, we will not be able to properly and effectively address global public health concerns like the pandemic just passed unless there are mechanisms in place that could advance epistemic justice, including the R&D work that we have heard a lot about yesterday and today, because ultimately these mechanisms need to be in place in order to advance the sort of trust and empower communities to effectively deal with shared challenges or collective challenges such as the pandemic. So to conclude, these are some points that I'd like to invite everyone to think about. Ultimately, how would the sort of data that we have currently speak to diversity concerns, and how ultimately the data sets that we have link back to social diversity, assuming that that's a good in itself. And if we think about it in terms of inclusivity, then there are a number of questions ultimately to consider within the genomic research space. So what should count exactly as an epistemic community in genomics? And who are the putative members that speak to ultimately the knowledge claims that are being generated? So ultimately, how does data represent both components that we considered above, and how are these two components ultimately interlinked? As I've mentioned, I think inclusiveness is an important bridge to a number of rights-based approaches. Of course, the right-based approach ultimately is one approach that will ground the pandemic treaty that is currently in discussion. And I do think that it will be a good opportunity for GA4GH to leverage on this particular point in supporting this very important global initiative that would, of course, ultimately feed back into how science is being practiced, including a number of funding and other related concerns that we've considered in the course of this particular conference. So thank you so much for your attention. look forward to learning more of your thoughts. Thanks again.
Excellent. Thank you, Calvin. It was a really important philosophical sort of foundation for a lot of the work we're talking about here today. So up next, it's my pleasure to introduce Laura Arbour, who is a professor in the Department of Medical Genetics at the University of British Columbia, an affiliate professor in the Division of Medical Sciences at the University of Victoria, both in Canada, and is a co-lead on the Silent Genomes Project, which we are very proud to support at Genome Canada. So Laura, the floor is yours.
Yeah, thanks so much. Oh, I guess you can hear me? Yes. Okay, so I really appreciate being invited here today. The discussions have been great. So many things that have already been mentioned this afternoon are relevant to what I'll be talking about, which is really a case study of what we are doing in Canada to ensure genomic inclusion to the extent possible for Indigenous populations. So we need to, as an overview, understand why it matters, how we are operationalizing all of this, and how we have, as Christian said, co-created governance so that this will be a sustainable entity. So I acknowledge with respect the Indigenous people of the lands that I live and work on, the Songhees, Esquimalt, and the Wasanich, and Coast Salish people. And I appreciate that we are currently on the unceded ancestral homeland of the Ramaytush, Ohlone people, who are the original inhabitants of the San Francisco Peninsula. So about 5% of the population of Canada is Indigenous. So who are the Indigenous peoples? So recognized by our government are the First Nations, and that includes over 1 million people, individuals in 634 different communities, and considered to be the first inhabitants, the original inhabitants of our lands. The Inuit followed about 4,000 to 1,000 years ago, and they inhabit, I guess you can't really see the red dots that they inhabit around the Arctic areas. 53 communities there, and Métis, or post-colonization since colonization of First Nations and European ancestry, are now recognized as a distinct Indigenous peoples. I wanted to mention that the Indigenous organizations, the Assembly of First Nations, the Inuit Tapiriit Kanatami, and the Métis National Council play a major role in the various processes that we have embarked on, and many others do. So there's great diversity with all of these communities, but there's great also challenges to healthcare, including genetic care. So I'm just going to walk you through some of the issues very quickly. You're going to get dizzy going through the slides very quickly, but I can provide them to you if you'd like more details. But really some important barriers to genomic diagnosis in healthcare, especially specialty care. We have geographic barriers, poor access to clinical expertise at the point of care, and additional barriers which we still need to continue working on, racism in healthcare, for example, long history of research transgressions, and the area that we're going to be talking about today, Indigenous populations are not represented in variant reference databases. So therefore, if they get to the table, they are not, have a less chance of getting a defined diagnosis. You've seen many different versions of the slides of the lack of increasing diversity in our reference databases. But for Indigenous people, what we see is a continued paucity or lack of that data. So why is that? Christian mentioned the importance of trust. People are not going to come forward. Many of these databases have been built from research, are not going to come forward if they are concerned about what will happen to them, what will happen to their data. So even in Canada with our residential school, shameful history of even nutritional research. This is not genetics or genomics. And how can you trust if pediatricians did this kind of research in conjunction with the government, and what will that mean even now? We have many stories to tell in this area. So many concerns that Indigenous people have expressed about genetics and genomics genetic causes may be stigmatizing. A variant may identify as a particular community, and I know that to be very true with the work that I do. Data may be used for exploitive purposes, secondary research without consent, mainstream benefit while health disparities continue. And this collage below is really about Indigenous people taking over and saying what is important to us, and how can we come together with a lens that actually considers what is important to us. So we do know that rare disease occurs in Indigenous populations just like in every other population. So we need to understand where we start with that. So we were fortunate to have an opportunity through Genome Canada, the large-scale applied research project, that allowed us to try to address this. But we needed to build that partnership. And this is, I'm going to be telling you how we did that. We decided we would build a governance model for use of Indigenous genomic data. We would offer, as a pilot study, genomic diagnosis to Indigenous children across Canada, which we did, and plan together how to build this Indigenous variant library. We had no idea at the beginning of the project whether that would actually happen. And then, of course, the importance of the economics. This diagram on the left really is about every patient needs to be put in touch with the appropriate healthcare provider. That healthcare provider simply needs to order the proper testing. We need to have the reference data to give the best chance of diagnosis for every child, every adult with a genetic condition. These are amazing funders. Thank you very much for those that are here. And this is the project that we built. Dr. Nadine Caron, one of our co-leads, a First Nation surgeon who came on board right from the very beginning. Dr. Wyeth Wasserman, it was actually his idea. He was involved with a postdoc, Dr. Maya Tarela-Gravik, who's actually at the University of Calgary, who said, this is an extremely important issue. We don't have the appropriate reference data. And we reached out to our international Indigenous colleagues, and this was, they played an extremely important role in advising us. Those are from New Zealand, from Australia, from the United States, and I always say from Hawaii separately. And then we had, actually once we were funded, we brought in a whole lot of folks internationally as well as nationally that were elders and community members and Indigenous organizations. And we all said, what do we need to do to get this started? And we came together for that. And we will talk specifically about the Indigenous Background Variant Library here. So we needed to build more alliances, and the Canadian Alliance for Healthy Hearts and Minds was already carrying out research and already storing samples for Indigenous under the guidance and leadership of Sonia Anand at McMaster University. A cohort study was being built to address cardiovascular disease and cancer. So I was working with them. We decided we would continue to connect with them. That might be the source. You can't just start this and expect 5 years later communities will come on board unless they've already been doing this for many, many years. And that's why we went to them. We got our expertise with genomics, population genomics, and Wyeth built an important team of folks. Last but not least, our community engagement coordinators, that we could not be able to do this without them. We, of course, build the infrastructure. Selene Currard built the pipeline. We also established we would have this background variant library at the BC Children's Hospital Research Institute. We assured that there was appropriate security. And then we started the community engagement. Brittany and I traveled across the country back and forth just pre-pandemic to visit these communities to say, is it okay that the stored samples could be used for this IBVL? So the process is a process that doesn't start with transferring the samples. It starts with community discussion and learning back and forth. We learn from them, they learn from us. And going through all of that, what we needed to do was establish a governance process. It's not enough to say, I promise. We need to have that governance process in place so that the trust is there, that we can continue on. And so this is where we developed our steering committee. And this steering committee was Indigenous people from across the country who had an interest either in the communities that they were involved with that may participate or had a genetic condition themselves. These were the tools that we had to build that kind of governance. And we had to ask the question, how can Indigenous data sovereignty coexist with clinician and patient access to genomic variant frequencies? So this is exactly what we were talking about so many times today. And I know I'm going over time, so 2 minutes. Because this is how do you, we can say that we're going to do this, but how do you do it? How can it coexist? And that's what I want to show you is how we believe that this can coexist and how it's being built. This steering committee came together 25 times to build all the processes that were important for publication, for use of the variants. It took us a year to establish how the variants would be used and released. I wanted to, let's see here. Yeah. The choice could have been absolutely open access like Nomad or very restricted, only certain access. And they chose, pretty close to restricted at the end, that there would be a manual registration, or there would be a registration with approval. Given that approval, they wanted these variants available internationally because they want to make sure that every child that has a diagnosis is diagnosed. So they were very clear about that, but they wanted to protect because they were trusting us, but there was the past to deal with. So an interface was built, and this is what we have ready to be released. And with that release, what we have is a statement of context that people actually understand that they're using this for the purpose of diagnosis. There are mechanisms for research, so if there is research to be done, that would go through another mechanism. But the agreement is that this is not to be used for publication, it's to be used for clinical diagnosis. And then the one-stop shop to actually show you what the variants are. With that, 600 samples were transferred, and they were transferred a few months ago. We are now sequencing, including long-read sequence and short-read sequencing, and the processing is underway. We are aiming for the first trials in 2024, including within the Silent Genomes Project, our pilot project. And this is a start. It needs to be sustainable. It needs to be expanded. But we believe that we are now headed towards what we were aiming for is the best chance of diagnosis. Thank you very much for that very fast, high-speed talk, and happy to give you the slides if you'd like more detail.
Wonderful, thanks, Laura. And just a phenomenal sort of assembly of talks that come together with philosophy, policy, and practice. So I think we have, if I'm not mistaken, something like 7 to 10 minutes for questions. So perhaps we'll get Slido up and people can ask questions, also vote early, vote often. We'll start here. Maili, you have a question to kick it off about how, if at all, the new diversity and data policy will align with recommendations from the most recent Academies of Science, Engineering, and Medicine report on using population labels in genetics.
Yeah, so I think we definitely went through the publication when it came out, and lots of the things anyway in the summary kind of overline about, you know, being really cognizant of the language that we use and kind of trying to understand better what we mean by certain things. And in there, anyway, it kind of makes that distinction between ancestry and genetic ancestry and provides examples for both that we've sort of alluded to within our own text. I don't know if you've both been involved in doing it as well, if you have any other thoughts.
Yeah, I know that Anna has integrated many of the dimensions of the NASEM report into the work and has kind of built on it and added additional dimensions. But no, a lot of that was incorporated into the report. Okay, excellent. Well, Alham, maybe we'll come back to you then. There's 2 questions. Maybe we'll put them together here.
First, at the bottom there, it asks about the impact that the lack of diversity and equity in biomedical research has had on health outcomes, drug development, and so on. And then maybe the corollary question is whether or not Broad's considered actually committing to minimum diversity baselines in data projects.
Yeah, I think I'll start with the first one, or the second one first. There's been no commitment for a baseline requirement, but there are institute-wide efforts that are really taking this seriously. So there's efforts in the Stanley Global Center, that are really thinking about how to build equitable partnerships to increase diversity in their data sets, specifically around psychiatric disease, and do that while also being involved in capacity building. So there's the efforts in the Stanley Center, and then the work of other scientists across the institute, such as Alicia Martin, that are really thinking about, and Hailiang Huang, that are really thinking about both how to increase the diversity of existing cohorts in terms of data sets, but also how to build new algorithms and models that can work with existing data sets and still extract meaningful data that is better able to make predictions for global populations and not, have the Eurocentric biases that we've had in the past. So I think there's kind of different ways that the Broad is trying to address this, both in terms of the research itself and kind of the algorithmic approaches to it, and then thinking about how to ethically build diversity and expand on diversity in various cohorts within the US and through all of us, and then globally via the Stanley Center, and other projects as well in Mexico and South America. And I forgot the first question already.
Yeah, that was just generally, I think, more about the ways in which the lack of diversity has impacted the delivery of positive health outcomes, whether that's through drug development or treatments and so on.
Yeah, I think, a lot of the previous talks have touched on the domains that you miss, right? Particularly thinking about rare variants and not capturing meaningful data from populations that are currently underrepresented in data sets. Thinking about kind of predictive capacity for polygenic risk scores. And that has been shown by Alicia Martin and several people that with existing Eurocentric bias data sets, the prediction levels are significantly reduced for non-European populations in terms of clinical predictability. So, the problems, I think, are well identified and defined. And I think, as a community, we're trying to think through them and work through solutions and be part of how to either get around existing underrepresentation in data sets or existing lack of diversity in data sets, or be directly involved in responsible cohort building that is thinking about more diversity in data sets.
Awesome. And it looks like we have a few minutes to spread out. So does anyone else want to pick up on any of that? We'll continue along, but we can expand on some of these questions, because I know that there will be different perspectives, perhaps from other panelists. But Laura, we'll turn maybe to you. There's a couple of questions here around Indigenous communities and genomics in particular. And I think, the second question on the list there, you maybe could speak to specifically about the work that Silent Genomes might be doing in collaboration with other initiatives globally looking at Indigenous genomics. But also, then maybe from that, we can go first to you and then to the panel on ways in which the GA4GH can actually help support responsible genomics with Indigenous communities or other underrepresented or marginalized communities. So, Laura, do you want to pick up maybe on the first question with regards to international work?
There are some discussions. I don't know that we've had Silent Genome specific discussions, but we know that the data sovereignty issues are extremely important to many of the Indigenous populations around the world. And this is, as we've expressed ours, is a freestanding database. And this is something that can be done effectively with other collaboration in the way that I'm not sure that combined data is going to happen, but each jurisdiction will likely hold their own, and there will be virtual agreements as to how there may be sharing. Yeah. And so when it comes to, and I'm going to jump into the second question just to start it, of course, for the other panel members. I do think that thinking outside the box is what we need to do. So we want to ensure that people are feeling welcome to come into the way we do things, but we also want to think about what is important to them and how can we think outside of the box and how can we meet them in the way that matters to them. And so it's really about us kind of changing the way that we do things so that others become, are willing and trusting and wanting to come forth. And that's going to be, I think, the answer.
I don't know if Maili or Calvin, if you want to pick up, of course, the work that that's already been done here with regards to the framework and policy, but ways in which that maybe can be interpreted with regards to some of the comments around Indigenous communities?
Yeah, I think there are some very good examples shared, even from this particular panel, and even more from what we've heard in the course of this particular plenary. I thought that just picking up on Laura's point about having governance mechanisms in place, I think these are some useful examples that could be fed up to some of the discussions at a global level. So I've mentioned the pandemic treaty that's in discussion. So one of the key components is to think about how exactly do we introduce such governance mechanisms that would ultimately speak to disadvantaged communities, including Indigenous communities, in order to empower them and to have them involved as co-producers of knowledge. Ultimately, this is what we want to do to ensure that they do have a stake in the sort of enterprise that's being introduced, so that ultimately this would be one of the key components to trust. So I think from a lot of discussions that we've had, it will be very helpful to bring this up to the global discussion and then to see how this could be perhaps considered for implementation elsewhere.
Great.
Thanks very much.
I was just going to say I completely agree with that. I think it's a lot about developing those relationships in us as the GA4GH community, being able to, empower and work together and figuring out ways that we can do that.
Excellent. Maybe we could clear a couple more questions off here because I think the top ones we've answered. I will just note though as well that from a funder perspective, I know that like Tiff and Catherine are here from Australian genomics, and there's a lot of work happening in Australia, as well as I know the NHGRI as well, and there's the learnings around working with Indigenous communities on Indigenous genomics certainly is a global conversation, and there's a lot of work and learnings that are going on also at that level as well. Wondering here, maybe take the question there on the bottom, I think is an interesting one for everyone, around any potential challenges or trade-offs associated with increasing diversity and representation around this kind of research. Maybe some of the challenges and barriers and ways in which that we're addressing that in the various projects you're working on. Who wants to kick off?
Yeah, I think I can start. I think one of the challenges is kind of related to aspects of my talk that because there's a lack of diversity, not just in data sets, but also in the scientists that are carrying out the work. There is not always the right people in the room when we're developing policies, when we're thinking about solutions. So really being intentional about finding ways to include people from communities, particularly communities, again, that have been underserved, excluded, or harmed, into these all of these conversations in terms of thinking about policies and kind of thinking about what the future might look like. I think that's something that I think about a lot, and I think it's just really important to be intentional about that with anything that we're doing.
Yeah, I'm just thinking about the policy itself. I think the question of benefit sharing and how to do that in a really responsible way, and how we can work together as the whole genomics community, whether that's through data sharing or helping with equitable collaborations and partnerships, whether that's providing funding, how to do that in a way that suits everybody is something that we spoke a lot about within that group, and I think is something that GA4GH could have more conversations about how to do that in a way that works for everybody globally.
Laura, do you want to pick up on that? Or maybe you can even reference this other question here around, which of course you mentioned in your talk, but I think you might want to emphasize that role of public trust and support.
Oh, public trust and support is huge. You know, at any given time, anything can happen that will erode that. So it is hugely important that we are listening and that we're talking and that there is a continuous conversation. And referring to the question above, you know, it takes time. It takes time to do this. It takes time, and it takes funders who appreciate how much time and the human resources that go into that. So that's the trade-off, is sometimes what we would be what we would consider efficiency and availability of all genomes. It's really coming together with that trust and to ensure that that we have our maximum opportunities.
Yeah, awesome. Calvin, maybe we'll take the question for you that's on here with regards to any specific actions or approaches that institutions like Global Alliance, but others as well, might take to ensure that epistemic justice is prioritized in the design and execution of the genomic research projects. So how do we put this into action?
Thank you so much. Great question there. I think what I'd like to see is if we have diversity in data sets in mind, then I think what might be helpful is to backtrack a bit, thinking about the data life cycle. The sort of selection criteria that we might have in place in terms of whom to include. I think this links up with what Alham has mentioned earlier on. So if we invite the same people ultimately to think about how a particular knowledge system is to be generated and advanced, then I'm not sure it ultimately would meet the cause of epistemic justice, because it just perpetuates the sort of injustices that we already find in the status quo. So it is very much, I think, with this policy, an invitation to, as Laura mentioned, to think outside of the box and to think about how we could have a more inclusive kind of system so that we could incorporate voices that are not otherwise in the system already, and so that we could then address some of the current understanding of who exactly has been included and how we are to understand marginalization and how we could advance knowledge in a way that is more collective and also in a way that could better engender trust. So, I do think that GA4GH has taken a great step forward in this direction, and now it's a matter of getting more account as to how some of the principles, some of the mechanisms discussed in the document could be put into action, and I think we've heard quite a number already. Yeah, thanks very much.
And we're starting to get towards the end, so maybe we'll take that the third question there, which is a bit of a pragmatic question with regards to bias, and in particular the ways in which you and your various projects maybe are actually defining questions for collecting demographics, social determinants of health data, other kinds of data to support greater diversity and inclusion, but while minimizing those biases. So maybe any experiences that you have in the way that you're approaching this would be useful to share. And I'm not sure who might want to kick that off.
I mean, I think we think about this a lot, and we're hoping to have, it's hard to kind of provide a checklist of like prescriptive things that one can do to avoid bias in kind of conceptualizing a research project or carrying it out or sharing it with the public. But I think one of the aims of the kind of research ethics consultancy is to have these conversations at the earliest stages of a project, and potentially if bias is part of the things that need to be considered, then you can address those domains at the earliest stages and maybe think of solutions to avoid bias and potential related harms. So that's the way that I kind of think about that. But
Maili?
Yeah, I'm trying to think within the group how much we discussed that. I think it's so difficult because can be so context dependent on what sorts of things were important. So we kind of put in the policy like a way to get researchers to start thinking about what the right way forward is for them and to think about their own context in defining kind of the collecting of the social demographics or social determinants of health that will support their own research. So instead of providing like one-all definition for people to use, we kind of tried to create a framework so people could think about those sorts of things themselves.
Great, thanks. Laura, anything from your side about kind of how you approach potential bias in your design?
Yeah, well, because right now we're working directly with First Nations communities. These are communities on reserve, and they self-identify as First Nations. So that in itself has not been an issue, but certainly this is something that we're addressing through our health authorities, because of course we are looking to share data, or not, we're looking to ensure that all have access and the appropriate defining populations need to be in there.
Excellent. Now, Colin, did you want to weigh in on this?
I thought that one approach may be to support interdisciplinary dialogue, because ultimately this could help to surface some of the biases that may be in place. Of course, we see in social science research, one of the difficulty with interdisciplinary dialogues would be to manage the conflicts that could potentially arise. Again, I do think that might be one approach, particularly if we do have, for example, social scientists, anthropologists in particular, that have worked with Indigenous communities for a long time. So they could perhaps serve as a conduit to think about some of the possible biases that could arise where Indigenous communities are concerned.
Okay, well, obviously there's a lot more to unpack here. There are still questions that we can get to, but perhaps the conversation can continue after the fact. Thank you for the important work you're doing. Please join me in thanking the panel for a super interesting discussion this afternoon.
Panel as well, and to Rob for chairing a great session. Thank you for doing it. Just to pick up on a few thoughts as I'm looking back at the day, and we haven't finished yet, so this is not closing the day. So let me just say, when we go to a break, and while you're having your coffee, please look at the posters, talk to each other. One of the recurring themes of GA4GH is how valuable the conversations are in addition to the presentations. So we're finishing a little bit earlier than we might do normally to give more of that time for conversations at your table, at the posters, in the coffee room. But just reflecting on the day, I think we started this morning look at some very big projects, some cutting edge technology, whether it was genomic technology or data science technology, and a real sense of how the power and the potential of those technologies to make a difference. We then went through sessions on how academic institutions, healthcare, and talk to industry, companies, small, large, economic development in a country, and that sense of a whole broad community. In GA4GH, about 50% of our member organizations are from industry, not just from academia or healthcare. And I think we've always had this sense that we needed everybody in the room together, not a sales exhibition outside and academics and healthcare talking science inside. So that sense of we need everybody in this room, everybody from industry to economic development to healthcare to all groups. And I think this session this afternoon has really pushed us into that recognition that when we say global, we really mean global, and the conditions, situations, opportunity, challenges that Christian described in what he had to do and his colleagues do to make a difference, not just in Africa, but for the world, and the challenges he faces there really set us up for this conversation now about the breadth of diversity and inclusion. And I can say that Tiffany's sitting here in the room. We're already talking about when we're in Melbourne a year from now for the next plenary, having a much stronger focus, particularly on Indigenous communities, and bringing people from all over the world to talk about this side of the issue. So it's been a great day of the breadth of GA4GH, from advanced technology, data science, all the way through the realities of inclusion and human interaction in order to bring the whole community together. So that's wrap up for today. I think tomorrow we've got another long day, which, remember that bit where you couldn't find the green button and I couldn't find my glasses? I still can't. We're going to start off with perhaps a voice we haven't heard too emphatically yet. Tomorrow morning, we've got a very dynamic speaker who's going to come at this from a patient perspective. So a parent and a patient perspective. He is. You will be left in no doubt about how important the work we all do is to families and people like him. We're then going to focus in on that third strategic imperative for GA4GH, this moving closer from research into healthcare, bridging the gaps, making the translation so that we can really make a difference for patients. So we'll start with hearing from a patient. We'll go through many of the challenges and opportunities in bridging from research to clinical. And then in the afternoon, we're going to get a keynote speaker from, Susan's going to talk to us from an NIH perspective, this huge impact that NIH can have, particularly in this country, but also globally. And so their data strategy is critically important, I think, to all of us. And then we'll have that conversation between the past and the future. And Heidi and Ewan will finish this meeting with sharing what it's been like to be a chair, what are the big challenges, the successes, and I think Heidi will take us where she hopes we all go in the next 5 years plus. So with that, thank you for the day. Look forward to seeing you tomorrow. But don't forget, coffee, talk to each other, look at the posters, talk to the presenters, and have a great evening. Thank you.